Lesson 7, Biology Lesson 7, Molecular Biology. Okay, Biology Lesson 7. We are talking about molecular biology. In this lesson, we are talking about molecules. Okay, molecules. Molecules means specially organic compounds, nuclear material. Okay, or genetic material. Nuclear material or genetic material. Okay, you can make your notes with me. We are talking about molecules. That means especially nuclear material. Nuclear material means some genetic material. There are two types of nucleic acids. Okay, nucleic acids. There are two types of nucleic acids. What are the two types? DNA and RNA. In most organisms, the genetic material is DNA, but in some viruses. Examples, influenza virus. Okay, influenza virus. Its genetic material is RNA. Okay. In most organisms, their genetic material is DNA. Therefore, in this lesson, we are mainly talking about DNA. DNA. Okay. DNA has special characters. DNA has special characters for acting as this genetic material. Okay, we call it as the vital genetic material. Vital genetic material in organisms. Okay, you can read your resource book. All the things I will mention all the things in your resource book. In your resource book. Okay. In first, you have to write the 4 characters. Okay, characters that are capable in facilitating DNA for acting as vital genetic material in organisms. Okay, there are 4 characters. What are the 4 characters? First one is accurate replication. Okay? Accurate replication of DNA. First one. Second one is transmission of genetic material from one generation to other. Okay? Transmission of genetic material from one generation to other. Third one, ability to store genetic material. Ability to store genetic material. Accurate replication, transmission of genetic material from generation to generation. Ability to store genetic material. How they store genetic material? As a nucleotide sequence. I will explain later. Fourth one is ability to express. Okay? Express heredity information. Okay? Ability to express hereditary information. Its hereditary information is transmit, store and express. Okay, DNA can transmit, store and express their genetic material, genetic information, heredity information, and it can also accurately replicate. Okay, those are the 4 characters that are capable in facilitating DNA for acting as vital genetic material in organisms. Okay, now we have to consider about the structure of DNA molecule. Okay, for describing the structure of DNA molecule, we are using some model. What is the model? What is the name of model? Watson and Crick model. No, Watson and Crick model. Watson-Crick model. Okay. As Watson, according to the Watson-Crick model, we know DNA has a double helix structure. DNA has the double helix structure. Okay. If it is a DNA molecule, there are hydrogen bonds. No? Okay, it has a double helix structure. No? When we magnify this small fraction of DNA molecule, we can see, okay, its monomer or building block. Okay, we can see its building block. What is the building block of nucleic acids? We called as nucleotide. Even in DNA or RNA, its monomer is called what? Nucleotide. In DNA, the nucleotide of the monomer is called deoxyribonucleotide. In RNA, its monomer is called ribonucleotide. That's the theory. Okay, in DNA, its monomer has 3 compartments. Okay, what are the 3 compartments? What are the 3 components? Okay, first one is deoxyribose sugar. That means pentose sugar. Pentose sugar means there should be 5 carbons. 1, 2, 3, 4 and 5. Okay, this is the deoxyribose sugar. Okay, just for memorizing. Okay, you have studied about all these things in lesson 2. Okay, chemical basis. Okay, in the chemical basis part, in the structure of DNA molecule. Okay, and other compartment is nitrogen base and phosphate group. This phosphate group is attached to the 5th carbon of this deoxyribose sugar. For one carbon molecule, this nitrogen base is added. Or nitrogen base is attached. Okay. In DNA or RNA, commonly called as nucleic acids, there are 2 types of nitrogen bases, purine and pyrimidine. Okay, purine and pyrimidine. Okay, in DNA there are 2 purine bases, adenine and guanine. Okay, in RNA there are 2 pyrimidine bases, thymine and cytosine. Okay, thymine and cytosine. But RNA, thymine is not present. Uracil is present instead of this thymine. Okay, uracil is only presented in RNA. Okay. In DNA molecule, it has a double helix structure. That means there are double strands in the molecule. These double strands are coiled as a clockwise ladder. Okay, ladder. That actually a twisted ladder. Okay, like that, twisted ladder. Okay. when we magnify this small fraction, we can see their nucleotides. Okay, 1, 2, 3, and 4. There are 4 nucleotides in this fragment. Okay. This DNA molecule has a double strand. Therefore, another antiparallel strand is there, like this. Okay. Sugar, 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 sugar. What is the name of sugar? Deoxyribose. Okay, this is the phosphate group. Phosphate group. Phosphate group means this is the fifth end of this strand, and this is the third end, and the 3 end. Okay, and this is the 3 end. This is the 5 end, because these strands are arranged in antiparallel way. Phosphate group. What are the two main bonds which are involving in this making of DNA molecule? What is the first one? Phosphodiester bond. Phosphodiester bond. Okay, phosphodiester bond is occurred between the phosphate group and 1, 2, 3. Third carbon of this the deoxyribose sugar. Okay, here same way. And there is another bond is involved. Those are the nitrogen bases. We have to consider about the nitrogen base pair role. Adenine is always paired with thymine. If there is an thymine base, okay, it should be adenine. Guanine, cytosine. Cytosine, guanine, like that. Okay. Between adenine and thymine bases, 2 hydrogen bonds are presented. Again, 2 hydrogen bonds are presented. 3 hydrogen bonds are presented. Okay. This phosphate group and this sugar group. Okay. We call it as sugar phosphate backbone. Sugar phosphate backbone. Here also a sugar phosphate backbone is present. Okay, sugar phosphate backbone. Okay, sugar phosphate backbone. Okay, we have to consider about the DNA double helix structure. There are some points. Okay, we can write the topic as DNA double helix structure. First point, who introduced this structure? Introduced by James Watson and Francis Crick. Second one is, what is the base of this structure? Okay, it is based on X-ray crystallography. Of DNA molecule. Who introduced or who obtained this X-ray crystallography of DNA? What is the name of this scientist? Rosalind Franklin. And the third one, what are the 3 main part of deoxyribonucleotide and what are the 6 molecules on it? Okay, what are the 6 molecules? we have mentioned there are three main parts, no? Deoxyribose sugar, phosphate group, and nitrogen base. So 1, 2, and 3 compartments are there. What are the 6 molecules? 1, 2, and what about others? Okay, in DNA, there are 4 types of nitrogen bases now: adenine, guanine, thymine, cytosine. Okay, adenine is a what? Nitrogen base. That means a molecule. 1, 2, 3, 4, 5, and 6. There are 6 molecules in this nucleotides. What are the 3 main parts? First one is deoxyribose sugar. Second one is phosphate group. Third one is nitrogen bases. 4 nitrogen bases. Okay. Purine, adenine and guanine, pyrimidine, thymine and cytosine. Fourth one, it is a twisted ladder. In a ladder, twisted ladder means like that, no? In a ladder, there are 2 compartments. Normally in a ladder, there are 2 compartments, no? What are the 2 compartments? This rails and this steps. Okay. In this DNA molecule, it is looks like a twisted ladder. Okay, twisted ladder, or also we can say spiral staircase. Okay, spiral staircase or twisted ladder. What are the rails? And what are the steps in this twisted ladder or spiral staircase in DNA molecule? In DNA molecule, this sugar phosphate backbone, act as a rail. Okay, rails are sugar phosphate backbone. What are the steps? These nitrogen bases and their hydrogen bonds. Nitrogen bases and their hydrogen bonds. Okay. And we have to consider about the nitrogen base pair rule. I have already mentioned about the nitrogen base pair rule. Fifth one is base pairing rule. Base pairing rule. There are 2 types of nitrogen bases, purine and pyrimidine. Okay, you can write those things. There are 2 types of nitrogen bases, purine and pyrimidine. What are the purine bases? Adenine and guanine. What are the pyrimidine bases? Thymine and cytosine. According to the base pairing rule, purine base is always pairing with purine. Is always paired with pyrimidine base. Okay, that means adenine is always paired with thymine, guanine is always paired with cytosine. There are 2 hydrogen bonds between adenine and thymine. There are 3 hydrogen bonds are presented between guanine and cytosine. Okay, you have to write all the things I have mentioned. And number 6, you have to mention about T.G. Morgan and his group. Okay, actually the experiments of T.G. Morgan and its group. Okay, especially their experiments. Okay, according to their experiments, they have introduced two statements. Actually, two statements. What is the first one? Chromosomes. Chromosomes are made up of DNA and protein. Okay, actually, in eukaryotic organisms, in their cells, chromosomes are presented. In the chromosomes, DNA is folded and bended. In one chromosome, only one DNA is presented. One chromosome, one DNA. Okay. The second thing is genes are the regions of DNA. Genes are the regions of DNA. Okay. Can you understand? In eukaryotic, especially in eukaryotic organisms. Especially in eukaryotic organisms. Okay, especially in eukaryotic organism, we are considering about a chromosome. In a chromosome, this is the structure of chromosome. Now, in a chromosome, DNA molecule is quite like this. This is the DNA molecule. Okay, this DNA, when we magnify this DNA, it looks like this. Okay, it has a double helix structure. It has two strands. It has two strands. In this DNA molecule, this DNA molecule, there are some regions. They are acting as genes. Genes. Okay, genes. Okay. Morgan and its group says genes are the regions of DNA. Okay, genes are the regions of DNA. There are also some regions which are not genes. Okay, non-gene regions are also presented in eukaryotic DNA. Okay, in this DNA is included into a chromosome. In a chromosome, it is called the centromere. Centromere, and it is a chromatid. It is also a chromatid and chromatid. Okay, and we can say this DNA strings make some genes. These DNA strings make some genes, and collection of these genes make chromosomes. When we consider about this analogy, okay, when we consider about the analogy, letters are the nucleotides. Okay, in a sentence, in this sentence, these letters are nucleotides. Okay, letters are nucleotides. Sentences means like, sorry, some some words means, or sentences are act as genes. Sentences. Some paragraph is act as a chromosome. Okay. This total collection is known as the genome. In this level, as a beginner, you have to think genome is some DNA collection. That's enough. Okay. Actually, the term genome is used for some collection of, for introducing some collection of genes. this all the set. Okay, this all the set. We can say this is the genome of some organism. Okay, in this level, okay, actually you are A-level student and you are beginner of this molecular biology. As a beginner, you can think the term genome. Okay, we use the term genome for introducing some DNA collection. That's enough for now. Okay. Okay. As I previously mentioned, this DNA is made up of some nucleotides. Each nucleotide is made up of three compartments: deoxyribose sugar, phosphate group, and nitrogen base. There are four nitrogen bases: adenine, guanine, thymine, and cytosine. Okay. When we comparing these four types of nucleotides in DNA, there are four types of nucleotides now. One, four types of nucleotides. That means the monomer. Okay, four types. Four types. Sugar, phosphate, phosphate. Okay, adenine, guanine, thymine, and cytosine. According to this diagram, you can see these bases make the differences between one nucleotide and another. Okay, actually the difference between this nucleotide and this nucleotide, what is the difference? This nucleotide has an adenine base, this has guanine base. Okay, that's the difference. Normally in this level you have to think about genes. Genes are controlling some characters. Okay, in the genetics lesson 6 we have discussed genes are controlling characters and alleles are controlling traits. No, okay, genes are controlling some characters. Genes are controlling some characters means for controlling characters, genes produce some proteins. Okay, some proteins. Genes produce some proteins. And actually, according to the specific sequence of this nitrogen bases, genes act separately. Okay, for each character. The information is in the order of nucleotide and the order is the information. Okay. Okay. Now we have to move on to the next. Now we are considering about chromosome, actually the architecture of chromosome. Architecture of chromosome. Architecture. What do you mean by architecture? Architecture of chromosome. Architecture means the way. Okay, what is the meaning of architecture? Architecture is the way of arranging. Okay, how it is arranged? What is the way of arranging of chromosome? Okay, okay. You can write the topic as architecture of chromosome. Architecture of the chromosome means the way of molecules. Molecule means DNA, like proteins, all the molecules are arranged in the well. We have studied the difference between prokaryotic and eukaryotic organism. What is the difference between prokaryotic and eukaryotic organism? What is the difference? What is the difference? Okay, for prokaryotic cell, we are considering about a bacterial cell. For eukaryotic cell, we are considering about an animal cell. In eukaryotic organisms, that means domain what? Domain eukarya. There are 4 kingdoms. What are the 4 kingdoms? Kingdom Animalia, Kingdom Plantae, Kingdom Protista, and Kingdom Fungi. In all those kingdoms, all the organisms have nucleus. Nucleus. They have nucleus means this is the nucleolus, and in eukaryotic cells their genetic material is enclosed by a nuclear membrane, and they have a organized nucleus, organized one, and they have a nuclear membrane, but they haven't. They have a circular DNA, we called as naked DNA. That means they are exposed. No nuclear membrane. Not organized. Okay, like that. That's the difference. Therefore, we can say their genetic material is included in the nucleus, but we cannot say in prokaryotic cell their genetic material, actually the circular DNA, is arranged in the nucleus. We cannot say like that. We can only say their nuclear material is arranged in the nuclear region. Okay, that's fine. Not the nucleus. Okay, we cannot use the term nucleus for these prokaryotic organisms. That is why we can say the architecture of chromosome means the way of molecules are arranged in the nucleus, right? Nucleus or nucleolus. What do you mean by nucleolus? That this is the nucleus. Okay, sorry, nucleolus. In bacterial cell, this is the circular DNA. Actually, this is the nucleoid. In the nuclear region, we call it as for the nuclear region we call it as the nucleoid. Okay, nucleoid, not nucleolus. What is the meaning of nucleolus in the eukaryotic cells? In the nucleus, nucleolus is presented. Okay. The architecture of chromosome means the way of molecules are arranged in the nucleus. So nucleoid or nuclear region of the cytoplasm. Okay, nucleus in eukaryotic cells, nucleoid in prokaryotic cell. Okay, eukaryotic cell and prokaryotic cell. In both, even the prokaryotic cells have only DNA, only circular DNA. They haven't a chromosome like in eukaryotic cells. Even they have no chromosome, no true chromosome. Commonly, we say in both eukaryotes and prokaryotes, DNA are called as chromosomes. Okay, DNA are called as chromosomes. You have to draw a table and divide it into two columns. In first column you have to write eukaryotic chromosome. We will study about the difference between eukaryotic chromosome and prokaryotic chromosome. Have you heard about histone proteins? Yes, no? Histone proteins are only presented in eukaryotic chromosomes. Even we called as chromosomes for both prokaryotic and eukaryotic DNA, the true chromosomes are presented only in eukaryotic chromosomes. They are false. Okay, their chromosomes are false. They have only DNA with the little amount of associated proteins. Okay, and they have several chromosomes. Some animal species have 2 chromosomes, 4. What is the number of chromosome in humans? 46. In human cell, there are 46 chromosomes are presented. That means 2 sets of chromosomes. That means 2 pairs of chromosomes. Okay. Anyway, 46 chromosomes are presented in eukaryotic organisms. More than 1 chromosomes are presented, but in prokaryotic organism, only 1 chromosome is present. Only 1 chromosome is present. Even 1 or 2 in each chromosome. Or in its chromosome. In the chromosome. Okay. In the chromosome or the circular DNA molecule, only one DNA molecule is presented. Okay, this DNA is eukaryotic DNA is linear, but prokaryotic DNA is circular. Look like this. Okay. In eukaryotic DNA is linear one, prokaryotic DNA is a circular one, and both have double strands and single DNA molecule. Each single DNA molecule, double strands are presented. Single DNA molecule. This DNA molecule is in eukaryotic organisms. This DNA molecule is associated with histone and other proteins. Okay, some complex proteins associated with histone proteins and other proteins. This DNA in prokaryotic cells, this protein is associated with the few protein molecules. Associated with the few protein molecules. Okay. Okay. As I previously mentioned, you have to think about the term genome is used for describing some DNA molecules, okay, some DNA sequences. The term genome, and we have talked about the architecture of chromosome. Now we have to consider about the uh, packaging of DNA in the nuclear region. Okay, DNA packaging. The DNA packaging. Packaging means the containment. Okay, containment. Containment of the genome, that means the DNA, containment of the genome in the nucleus or nucleoid. Okay, nucleus in eukaryotes, nucleoid means prokaryote. Okay, DNA packaging means the containment of genome in the nucleus or nucleoid. That's the DNA packaging. You can write the containment of the genome or DNA in the nucleus or in the nucleoid. Okay, nucleoid is called DNA packaging. I have mentioned in a chromosome DNA is packed inside. Okay, how DNA is packed inside in this chromosome? Okay, what about the difference between size of the chromosome and size of DNA? Okay, when we consider the size of all chromosome in an organism, there is an enormous DNA enormous amount of DNA is included in all chromosome of an organism. Okay. Anyway, how DNA is packed? Okay, how it is contained? Is associated proteins are helping. Okay, these associated proteins are helped in the DNA packaging. Okay, we have to consider how these associated proteins help in the DNA packaging in both eukaryotic and prokaryotic cells separately. In first, we are talking about prokaryotic DNA packaging. Prokaryotic DNA packaging. Okay, prokaryotic DNA packaging. In prokaryotic DNA packaging, we are talking about the packaging of DNA. Okay, this packaging is facilitated. This packaging is facilitated by their DNA associated proteins. This packaging of DNA is facilitated by their associated proteins. Okay, how it can be done? How it can be done? For packaging DNA inside this chromosome or inside the nucleus in eukaryotes, these facilitated proteins are involved. How these facilitated proteins are helping? How? How? These these proteins cause the DNA to coil and supercoil. Coil is also known as fold or some loops. Okay, both terms can be used for coil. The term coil. These proteins cause the DNA to coil or supercoil. Then DNA can compact into the nucleoid or nucleus. Okay, that's the reason. In this part, we are talking about the prokaryotic DNA packaging. Now, therefore, we can say this associated proteins cause DNA to coil and supercoil. Then DNA can compact into the nucleoid. Okay, nucleoid, not the nucleus. Okay, because we are talking about the prokaryotic DNA packaging. Okay. In prokaryotic DNA packaging, normally in the bacterial cell, it has a circular DNA. This is the circular DNA. Okay, this is the circular DNA. Okay, what is the magnified diagram of this DNA? Even we draw it in a single line, its actual structure is like this. Okay, double strand, single circular molecule. Okay, it looks like this. There are hydrogen bonds between two strands. No? This is the actual structure of prokaryotic DNA molecule. Okay. If it is a DNA molecule, that means circular unfolded. This term unfolded you will understand in future. Okay. This is the circular unfolded chromosome. So, chromosome in prokaryote, actually this is not a true chromosome, actually this is a DNA molecule with the associated protein. This is the circular unfolded chromosome. For packaging with the help of these associated proteins, this circular unfolded chromosome, unfolded chromosome initially coiled into loops or folds like this. Okay, initially coiled. Those are called as loops or folds. Loops or folds. Okay. In prokaryotic organism, in prokaryotic DNA, I have mentioned there are associated proteins are also presented. These associated proteins are presented in here. Okay, that is the core. It is composed of RNA and protein. Okay, in the circular unfolded chromosome initially coiled into loops or also called as folds. What is the reason for this coiling? What is the reason? For packing. For packing. Okay. For packing. This is the stage 2. Diagram number B. Then what happened? Each loop independently coil. Independently coil. That means this each coil is independently supercoiled. Okay. Each loop or each fold is each loop or each coil is independently supercoil of a supercoil. It looks like this. Those are the supercoils. So those are the supercoils. Supercoils. This is the RNA protein core. RNA protein core. Okay. These supercoils also known as domains. Okay, domains. These supercoils also known as domains. Domains. Okay, domains. These supercoils or domains also known, we can say, loops of compact mass of DNA. Loops of compact mass of DNA. These supercoil or domains also known as loops of compact mass of DNA. Okay. These mass of DNA or these supercoils or domains are bound to a core. Okay. Bound to a core. Bound to a core. Okay. This core is made up of RNA and protein. Okay, RNA and protein. Okay, this core is attached to the membrane. Okay, this core is attached to the chromosome. The chromosome to membrane. That is why these chromosomes are packed or contained in the nuclear region. We are talking about prokaryotic chromosome. Actually, DNA, the circular DNA and its associated proteins. Normally the DNA is look like this. For packing, their associated proteins are helped in the initial step, this circular unfolded chromosome. Okay, now you can understand what is the meaning of unfolded. Okay, in here this chain, this circular DNA is folded. Okay, that means folds or loops are created. Okay, after folding. Therefore, the initial stage, this circular DNA, this circular chromosome is an unfolded one. Okay, for packing with the help of associated proteins, initially coiled into loops or folds, and each loop or coil is independently supercoiled. These supercoils are also called as domains, and also it's called as loops of compact mass of DNA. These supercoils or domains are bound to a core. This core is made up of RNA and protein. This core is bound to a membrane. That is why DNA is packing. DNA is packing in the nuclear region of prokaryotic organism. Okay. I will erase this. Please hurry up. Hurry. This loop or fold independently supercoil and make domains of supercoils. These supercoils, okay, this supercoil also can be relaxed. Okay, also can be relaxed by introducing a process called as single strand mix. Okay, single strand mix. Okay, and now we have known in the prokaryotic cell for packing inside the nuclear region, DNA is supercoiled and make domains, and it is bound to a protein RNA core. This RNA core is attached to a membrane. Therefore, DNA can pack inside the nuclear region. And after bounding this core with the membrane, there is a barrier to rotate. There is a barrier to DNA molecule for rotating. Okay, barrier for rotating. Barrier for rotating. Barrier for rotating of the chromosome. Barrier for rotating of the chromosome. And this is how we can explain this. We can write, since the chromosomes are attached to the membrane. Since the chromosomes are attached to the membrane, and held together by the core, which is made up of RNA and protein, no? Held together by the core, acting as a barrier for rotating. Okay, that's the region. Since the chromosomes are attached and held together by a core, then the chromosome cannot rotate. Okay, that means acting as a barrier for rotation. Okay, and there is another point. We have already discussed about this part. Domains or supercoils can relax into loops. Okay. What is the importance? What is the importance of domains are relaxed, or actually this is, if it is a forward reaction, supercoils are become loops. This is the forward reaction. Okay, like an equilibrium. Okay, what is the importance of this one? Okay, these supercoils or domains can relax into loops, and again it can supercoil into domains. What is the importance? Important is you have to write a specific term. Transcription of What is the importance of this process? Okay, forward reaction and backward reaction. Important is transcription of specific genes. Transcription of specific genes. And how we can remove this or after removing RNA molecules. That means in supercoils they are attached to a core. This core is made up of RNA and proteins. Okay. If we remove RNA, removal of RNA will lead to loss of independence of the loops. Okay, loss of independence of the loops. Okay, removal of RNA. Removal of RNA lead to loss of independence of the loops. And we are talking about prokaryotic chromosomes now. They are chromosomes. Actually, this is the DNA with associated small proteins. Okay. In prokaryotic genetic material, mainly the nucleoid or the main circular DNA. There is another genetic materials are there, we called as plasmids. These plasmids are using in gene engineering technology for producing genetically modified organisms. Actually, beneficial organisms. Okay, genetically modified for producing genetically modified organisms. This is the main DNA. Okay, circular DNA. Main circular DNA. In some bacteria, some prokaryotic organisms, another genetic material, plasmids. Plasmids are presented. This is the structure that always be presented. This is the structure of sometimes presented. Okay, sometimes presented. In all bacterial cell, in all prokaryotic cell, definitely this main circular DNA should be there. Okay, but these plasmids, we can see these plasmids only in some bacteria or some prokaryotic organisms. These plasmids we known as extra chromosomal. Okay, extra chromosomal genetic elements. These plasmids also can be coiled or supercoiled. Okay, these plasmids also can be coiled or supercoiled. These plasmids are called as extra chromosomal genetic elements. Okay, only present in some bacteria or some prokaryotic organisms. Okay, that's all about the packing, DNA packing of prokaryotic organisms or prokaryotic DNA packing. Okay. Now we have to consider about the eukaryotic DNA packing. Okay. Now we have to consider about the eukaryotic DNA packing. Okay, now we are talking about the eukaryotic DNA packing. Eukaryotic DNA packing. Uh, as I previously mentioned, these eukaryotic organisms have the complex protein called what? Histone proteins. Okay, in their cells, chromatin is present. Okay, this chromatin is made up of DNA and protein. Okay, how it is produced? This DNA and protein are involved in the formation of chromatin. Okay, this DNA is associated. This DNA is associated with a large number of proteins, specially called histone proteins. Histones. Okay, these proteins. Okay, these histone proteins help to organize the DNA inside the nucleus. Okay, here we can directly say in the nucleus. Okay, not the nucleolus, because we are considering about the eukaryotic DNA packaging. In all eukaryotes, the nucleus is present in their cell. Okay, this chromatin. Okay, this chromatin, which is composed of DNA and protein. There are two types of chromatin. First one is euchromatin. Second one is heterochromatin. Okay, in the nucleus or in the cell, in eukaryotic cell, the nucleus is present. This is the nucleolus and chromatin. There are two types of chromatin. Okay, like this. Okay, one type is compactly packed and other is loosely packed. Okay, that's the difference between these two types of chromatin. The lightly packed chromatin is called, okay, this one. The lightly packed chromatin is called euchromatin. Okay, that means it is lightly packed. This one, another one is called heterochromatin. It is tightly packed. Okay. There is another information about euchromatin. It is rich in genes. Okay, rich in gene means it is probably active in transcription. Okay, transcription in the protein synthesis. In here, they haven't probably active genes, but they are also chromatins. Chromatin means it is made up of DNA and protein. Its DNA is composed of nucleotides now. So in heterochromatin nucleotides, their nucleotides sequence mostly inactive. Okay, even its nucleotide sequence is mostly inactive, there are some advantages. That is why heterochromatins are presented in the nucleus. Okay, there are there are two types of chromatin, euchromatin and heterochromatin. Euchromatin is lightly packed while heterochromatin is tightly packed. In euchromatin, it is rich in genes, that means probably active in the transcription in protein synthesis. But in here, in heterochromatin, their nucleotide sequence is mostly inactive, but there are some another advantages. There are three advantages. First one is gene regulation. Their genes are regulated by this heterochromatin. Okay, genes regulation. Second one, epigenetic inheritance. We have previously discussed about this topic, epigenetic inheritance, in lesson 6. Okay, that is the genetics lesson. Third one is protection from chromosomal Okay, all the things that I have discussed, all the things in your resource book, please refer it, okay, and make your notes, complete your notes. Okay, then we will talk about the formation of DNA as the chromatin or as the chromosome. The final aim is packing, DNA packing in the nucleus. In eukaryotic cell, there are 4 steps for producing chromatid or chromosomes actually. Okay, step 1, step 2, 3, and 4. Can you remember about the prokaryotic DNA packing? The circular DNA, there are some loops, and each loop is independently supercoiled and produce domains. These domains are associated with the protein RNA core. This core is attached to the membrane. That is how it is packed, okay, in the nuclear region. And in this prokaryotic DNA, there are 4 steps, 4 levels, okay, or 4 levels. In the level 1, the special protein that we have discussed previously, histone proteins, the 8 histone protein molecules are combined. 8, okay, 1, 2, 3, 4, and another 4 histone proteins are behind this one. Okay, this looks like a cubic. Okay, like that. Okay, there are 8 histone proteins are collected, and the double helix of DNA winds it. Okay, winds around the 8 histone protein molecules. Okay, the diameter of this structure is about 10 nanometer. In the level 1, this one has double helix structure like that. Okay, this double helix structure winds around a complex of 8 histone molecules like that. Okay, 8 histone molecules. Again, there are 8 histone molecules, and the double strand DNA is brings around this, like that. Okay, and this each unit is called nucleosome. Each unit is called nucleosome, and these joining DNA fragments are called linked DNA. Okay. These nucleosomes and their linked DNA looks like a bits of a necklace. Okay, bits of a necklace. Bits of a necklace. Okay, these units are nucleosomes and they are linked DNA. Linked DNA. Okay, this double helix structure wings around a complex of eight histone protein molecule. These are called, each unit is called nucleosomes, and they looks like a bits of a necklace. And when we considering about the linked DNA, the adjoining beds of nucleosomes are linked together by a stretch of DNA is called the linked DNA. Okay. In step 1, the special name is the nucleosome unit. Okay, nucleosome unit. In step 2, uh, we can represent each nucleosome like this. Each nucleosome like this. Okay. In step 2, what happened? This nucleosome twist, okay, twist and pack in a spiral fashion, and this 30 nanometer diameter fiber is called chromatin. Okay, chromatin. Okay, this 30 nanometer fiber is called chromatin. The special name in the step 2 is chromatin. Chromatin. Those are chromatin fibers. Okay. In the second level, these nucleosomes twist and pack in a spiral fashion. Pack in a spiral fashion and form a chromatin fiber. Form a chromatin fiber. Okay. It is roughly in 30 nanometer diameter. And in step 3, what happened? In step 3, this chromatin fiber, 30 nanometer chromatin fiber forms loops like this. Okay. This chromatin, okay, 30 nanometer diameter chromatin fiber forms loops like this. Loops like this. Okay. These are called loop domains. Okay, the loop domains. The diameter is about 300 nanometer, and it is attached to a protein scaffold. Okay, protein scaffold. It is attached to a protein scaffold. In the level 3, this 30 nanometer fiber forms loops like this, and these loops are called loop domains, and the loop domains attached to a protein scaffold. Okay. In the level 4, these loop domains are coiled again, and this protein scaffold is also bending, or it is folding. Okay, like this. This one is folding like this, and each loop domains are coiled independently like this. Step 4. Okay. Here the diameter is about 700 nanometer diameter. Okay. And from this structure, again, the with the help of the nuclear matrix, again another scaffold is produced, and with the help of nuclear matrix, the chromosome is produced like this. Okay, those are the sister chromatids, centrosome. The whole molecule is called chromosome. Okay, whole structure is called chromosome. It is about 300 nanometer diameter. Okay, it is actually a metaphase chromosome. That's all about the DNA packing in both prokaryotic and eukaryotic organisms. Now we are moving into the DNA replication theory. It is very important. So I will stop for now.